Good day. Today we're covering two other forms of nutritional anemias. Other than the iron deficiency anemia, there are two other forms, which are folic acid and vitamin B12 deficiency anemia. And in contrast to iron deficiency anemia, these two forms cause what we call macrocytic anemias. And in particular, with folate and vitamin B12, they are megaloblastic anemias. And we'll talk about the difference between megaloblastic and non-megaloblastic macrocytic anemias in just a while. So by definition, a macrocytic anemia is anemia where the mean corpuscular volume is large in contrast to microcytic. It's above 95 femtoliters. So when you see large RBCs in the periphery with anemia, it means this is macrocytic. Now, this can be megaloblastic or non-megaloblastic. The difference between them is that megaloblastic anemia is one which is associated with abnormal DNA synthesis in the bone marrow. So the macrocytosis or the enlargement occurs also in the bone marrow, and you can see megaloblastic changes in the bone marrow. In contrast, non-megaloblastic and macrocytic anemia is not due to interference with DNA synthesis, and it is associated with certain diseases that cause macrocytic changes in the circulating RBCs. These diseases include things like liver disease, like alcoholism, although alcoholism can also cause megaloblastic changes sometimes. Hypothyroidism can also cause it. So many other diseases and some drugs as well. So our discussion today is focused on folic acid and B12, which cause megaloblastic anemia. And as we said, this is a disorder of impaired DNA synthesis. And it doesn't only affect the RBCs, but it also affects other cells in the bone marrow itself and in other sites in the body. So what happens? The interference with DNA synthesis results in a delay in the maturation of the nucleus, while the cytoplasm continues to grow. And as a result, you get these large and frequently dysfunctional cells. The RBCs are larger than normal, and the neutrophils typically in this situation will have what is called hypersegmented nuclei. Normally, the neutrophils have segmented nuclei, but in this case, they are hypersegmented, having more than 5 or 6 lobes in the nucleus. So most often this megaloblastic anemia is due to B12 or folic acid deficiency, although there are other rare causes. And this is a typical blood smear of a normal person. You notice the RBCs, which are round, uniform in size, relatively small compared to the neutrophils and the lymphocytes and the monocytes. Contrast this with what happens in megaloblastic anemia, and you can see that in some cases, at least here this is an RBC compared to a neutrophil. Obviously, this is a much larger RBC. You can see it also on the right panel. This is a lymphocyte, and the RBCs are much larger. And again, here below, this is a macrocyte. But also, you might see different sizes, cytosis, small, large, even abnormally shaped cells like these teardrop shapes here, this one and this one. And this is what you see in a state of megaloblastosis. And this is a typical hypersegmented neutrophil that is associated with megaloblastic anemia. In the bone marrow, again, you see these similar large cells, the megaloblastic changes, and the erythroblasts. You can see these fine stippled nuclei, the chromatin, abnormal development. I'm sorry I missed it, but you can also see in the lower slide, on the right, the metamyelocytes, which are also enlarged. So what is the basis for this anemia? Both vitamin B12 and folic. There is a relationship between the two. And the basic problem is the inability to convert uridylate to thymidylate, which is essential for the synthesis of DNA. So you're interfering with DNA synthesis by blocking or not allowing this step to happen. And this is a step that requires a certain form of folic acid called 5,10-methylene tetrahydrofolate polyglutamate. And you've studied this before in studying the drugs like methotrexate, sulfonamides, trimethoprim, all of which interfere with folic acid metabolism and eventually are going to block this step in the synthesis of DNA. So this deficiency in ability to convert uridylate to thymidylate can result from either folic acid deficiency or vitamin B12 deficiency. Now, this busy slide, don't be afraid of it, summarizes the metabolism of vitamin B12 and folic acid and the relationship between them, and identifies where the defect when you have deficiency, where the defect occurs. So vitamin B12 metabolism is here in A. Folic acid is in B and C, and the site where the defect occurs is here, where you get conversion of uridylate to thymidylate, which then goes into DNA synthesis. And this is the role of 5,10-methylene tetrahydrofolate polyglutamate, which is essential for this conversion. Okay, so we're going to focus on A and B. Remember that folic acid is also involved in purine metabolism and histidine metabolism, but we're not going to dwell on this during this lecture. So this is the legend for the previous figure. You can read it later on. I'm going to explain it in just a while. So here, let's focus on vitamin B12. So this is vitamin B12, also known as cobalamin. This is how you take it in through food. And there's a process for absorption and so on. We'll talk about it later. But basically, vitamin B12 has one of two fates. It's either converted to deoxyadenosyl vitamin B12, which converts L-methylmalonyl, or is essential for converting methylmalonyl to succinyl-CoA, which then goes into the Krebs cycle and has other roles, as you know. Or the cobalamin, vitamin B12, can get converted to the methyl form. And this methyl form is important because it, together with homocysteine, results in the formation of methionine and then S-adenosyl methionine, which is important in the synthesis of many compounds in the body: DNA, myelin, amines, and proteins. And so you can imagine that deficiency of vitamin B12 is going to result in deficiency of SAM, S-adenosyl methionine, and is going to have multiple consequences. Now, how does vitamin B12 get converted to methyl vitamin B12? It is through a donation of a methyl group from the methyl tetrahydrofolate to vitamin B12. The methyl tetrahydrofolate then becomes tetrahydrofolate. And this tetrahydrofolate is what eventually gets converted to the active and storage forms of tetrahydrofolate. And so tetrahydrofolate becomes polyglutamated, as you say. See here, tetrahydrofolate N, it means adding multiple glutamate groups to the tetrahydrofolate molecule. And this is the precursor of the active compound which we are concerned with here, which is 5,10-methylene tetrahydrofolate. So when you have a deficiency of folic acid, which is the methyl tetrahydrofolate we take in food, we get a deficiency of 5,10-methylene tetrahydrofolate. As a result, you get decreased conversion of UMP to TMP and decreased DNA synthesis, and that's the cause of megaloblastic anemia. By the same token, if you have a deficiency of vitamin B12, this again will prevent the conversion of methyl tetrahydrofolate to tetrahydrofolate, which is the precursor of tetrahydrofolate here. So either the deficiency of vitamin B12 or the deficiency of folic acid is going to interfere with the formation of 5,10-methylene tetrahydrofolate. And both of them are going to cause megaloblastic changes, megaloblastic anemia. Importantly also, during vitamin B12 deficiency, what happens is there is an activation of a reductase, a 5,10-methylene tetrahydrofolate reductase here, that converts this compound to methyl tetrahydrofolate. It sort of shunts it there, preventing it from participating in this. So what you're doing is trapping folic acid in the form of methyl tetrahydrofolate. But this methyl tetrahydrofolate cannot form tetrahydrofolate because you have a deficiency of vitamin B12. And so both deficiencies eventually are going to cause megaloblastic anemia. So these are some of the points I mentioned. The methyl tetrahydrofolate is the form that is supplied to cells. This is the one which donates methyl to vitamin B12. Tetrahydrofolate is the precursor of the polyglutamate tetrahydrofolate. This is the one which participates in the metabolic activities of folic acid. And the 5,10-methylene tetrahydrofolate is the one which is of concern for us now, which converts uridylate to thymidylate. So in folate or B12 deficiency, as we said, cells redirect the folic acid metabolism by activating this reductase, which results in the formation of methyl tetrahydrofolate. So let us discuss now vitamin B12, its absorption, handling, and results of consequences of its deficiency. Vitamin B12 is produced by intestinal microorganisms in animals, not in humans, however. So it's available to humans in animal products, not in vegetables, not in legumes, unless in certain circumstances where they are contaminated by bacteria that can synthesize it. In other words, people who are vegetarians are at risk of developing vitamin B12 deficiency and must receive supplements if they don't take in any animal products. This is vitamin B12. Again, you have here a porphyrin-like molecule with a central cobalt atom instead of iron, which is in heme. And there's a nucleotide linked to the cobalt as well as a methyl or another group that is linked to the cobalt from the other side. It could be the methyl, which is in methyl vitamin B12, or it could be the 5-deoxyadenosyl group, which participates in the conversion of malonate to succinate. Now, how is it absorbed? Vitamin B12 is taken in the diet and goes to the stomach. In the stomach, it is susceptible to acid degradation, but it is protected from this by a protein secreted there called haptocorin, also called transcobalamin 1. So it binds to this protein and bypasses the stomach, reaches the intestine. In the intestines, it combines now with a factor called intrinsic factor here, which is also secreted in the stomach from the parietal cells. The intrinsic factor travels to the intestine, meets the vitamin B12 there, and binds it. They travel together to the site of absorption of vitamin B12, which is in the ileum, the distal part of the small intestine. There is a special receptor for intrinsic factor, which results in the absorption of this complex. And this complex then is handled, and we'll discuss it in the next slide, but just as a general idea here, B12 is absorbed, it's taken to the liver, the liver may recirculate it to the body. It can bypass the liver and go to the body, or it can be secreted again, and it can undergo an enterohepatic circulation. So secreted, the bile reabsorbed in the intestine. And here is what happens in the body. So let's start again from the intestinal cells here. This is intrinsic factor linked to vitamin B12 or cobalamin. It meets the receptor for intrinsic factor, which is called tubulin, which is linked to another protein. No, don't worry about this. This absorption takes place through the internalization of these molecules. The intestinal cells degrades intrinsic factor and releases cobalamin. Cobalamin then binds to a protein called transcobalamin 2, and this is released into the circulation as transcobalamin 2 cobalamin complex. Now, also some of the cobalamin may be bound to haptocorin itself and circulate as such. There are also analogs, cobalamin A of cobalamin, don't worry too much about this. But in any case, the cobalamin, which is bound to either haptocortin or to transcobalamin 2, will circulate in the body. Transcobalamin 2 is the one which delivers cobalamin to the cells that need it, such as the one in pink on the right here. So there is a transcobalamin 2 receptor which binds this complex and internalizes it. And again, transcobalamin 2 is degraded or recirculated, and cobalamin is then used in the various metabolic processes, whether through conversion to methyl or deoxyadenosylcobalamin. cobalamin. In addition, this cobalamin may go to the liver either as transcobalamin 2, which gets absorbed the same way, or through the haptocorin cobalamin, which gets absorbed through another receptor, SGP receptor, and then cobalamin is released from the receptors. It can be stored in various forms. One of them is binding to a protein called transcobalamin 3 or to enzymes, and also it can be secreted into the intestine. It can be secreted as free cobalamin or haptocorin-bound cobalamin. In either case, the haptocorin is degraded by pancreatic enzymes, and you release free cobalamin, which gets bound to intrinsic factor, and as such can be reabsorbed, as we said earlier, in the intestine, or what is not reabsorbed gets eliminated in the feces. So this is the story of cobalamin, how it's taken up, distributed to the tissues, and stored in the liver, and the important role of certain proteins in this pathway. So there are three transcobalamins, 1, 2, and 3. 1 is haptocorin, 3 is form of storage, and 2, transcobalamin 2, is the one which delivers cobalamin to the cells where it is needed, because there is a receptor for it that takes it up. And this is the explanation of the previous figure. So, to reiterate, vitamin B12 absorption happens by binding to haptocorin in the stomach to protect it from acid. It is released from this in the duodenum. The intrinsic factor binds it. In the ileum, it is reabsorbed by binding to a receptor called tubulin. And then in the after absorption and release, it is bound to transcobalamin 2 and transported to cells. The transcobalamin 2, vitamin B12 is taken up by tissues and vitamin B12 is released. Now, the storage of vitamin B12 in the body, stores can range from 1 to 10 milligrams, usually higher, on the higher side. The requirements, therefore, for vitamin B12 to function and to maintain the stores are approximately 2.4 micrograms per day, or 877 micrograms per year. These are the requirements for utilization. In other words, if you have a 10 milligram store of vitamin B12 and you need 877 micrograms per year, your stores will range maximum around 11 years. In other words, if you stop taking vitamin B12 now, you can sustain yourself for a number of years, depending on how much your stores are. And the deficiency symptoms will start after the stores are depleted. Now, when you have things that interfere with the enterohepatic circulation, like surgeries, gastric surgery, where you don't have intrinsic factor anymore being released, this will shorten the period from deficiency to appearance of the symptoms. So the stores will not last as long because you're losing more vitamin B12. What can cause vitamin B12 deficiency? Anything that interferes with this process. Obviously, depletion in the food intake. If you don't have intrinsic factor, which is essential for its absorption, and this can happen in certain cases. For example, there is a particular disease called pernicious anemia, which is associated with antibodies to intrinsic factor and to certain cells in the stomach mucosa. In addition, if you have problems with absorption at the ileum, at the level of the ileum, such as if you have surgery to remove the ileum due to inflammatory diseases of the bowel, Crohn's disease is one, or other diseases called tropical sprue. You'll study these in the GI course. There could be absence of transcobalamin 2, the protein that is necessary to deliver vitamin B12 to the cells. As we said, gastric surgery, which interferes with reabsorption and recirculation, can eventually result in faster depletion of the stores. And when you have elevation of TC1 and TC3, these are the proteins that can bind vitamin B12, but when they're elevated in the blood in certain diseases, what they do is bind this vitamin B12. However, this bound B12 cannot be made available to the cells. The way it's available to the cells is by binding to TC2, transcobalamin 2, which has the receptors on the cells to internalize it. So this becomes a state of practically its deficiency of the cells, for the cells. Pernicious anemia, as we said, is a special kind of anemia that is associated with antibodies to either intrinsic factor itself, but also to parietal cells components like the hydrogen pump that is responsible for secretion of hydrogen. And so, in addition to the anemia, you will see achlorhydria, in other words, decreased or no secretion of acid into the stomach. There's no intrinsic factor, there's atrophy of the mucosa, and you can detect antibodies, both to intrinsic factor, which therefore cannot bind B12 anymore. What are the signs of symptoms of megaloblastic anemia due to vitamin B12? Well, of course, we're not going to reiterate the signs of symptoms of anemia. We know them. They're the same as for iron deficiency anemia. That's the hematological symptoms: tiredness, dyspnea, increased cardiac output, etc. But especially for vitamin B12 deficiency, there are other problems that arise, particularly neurological consequences. And these, if left untreated, can be irreversible. What you see is pathology. On pathology is swelling, demyelination, cell death in the spinal columns and the cortex. And symptoms include sensory changes, paresthesias, particularly in the lower extremities, imbalance, ataxia, confusion, and even psychotic symptoms. Optic atrophy can happen. And exact reason for this is not known. It is thought to be related to the participation of the methyl vitamin B12, which is not being produced now, in the formation of S-adenylyl methionine, and we said that is important for synthesis of very important compounds, proteins, including myelin and others. So when you want to treat vitamin B12 deficiency, you have to be as specific as possible, obviously. You no need to use multiple vitamins. Try to target the vitamin that is deficient, which is vitamin B12. And you have preparations of vitamin B12. It could be oral, and it could be parenteral or intramuscular or subcutaneous. Now, as you can tell from the causes of vitamin B12 deficiency, unless it is decreased intake in the diet, which you can treat with oral supplements, all the rest involve abnormal absorption, inability to absorb vitamin B12. So in those situations, you obviously cannot treat with oral preparation of vitamin B12, and you have to resort to parenteral administration. If there is severe pancytopenia, there's severe neurological deficit, and particularly if there's bleeding, you have to probably give IM injections and might require blood transfusions, particularly if there's bleeding. And again, as we said with iron deficiency anemia, you should monitor the response once you start therapy. If the patient does not respond as expected, then this suggests there might be other deficiencies. It might not be an isolated vitamin B12 deficiency. There could be also folic acid deficiency, or there could be iron deficiency as well. Or there might be other diseases that are causing anemia, such as inflammation, chronic disease, anemia of chronic disease, or renal disease, which is associated with decreased erythropoietin production by the kidney. So moving on to folic acid, we'll do the same as for vitamin B12. There are many forms of folic acid, as you will see in a while. This basic structure of folic acid is pteroyl glutamic acid, which is formed of a pteroic acid combined with glutamic acid, as the name implies. Pteroic acid is a combination of para-aminobenzoic acid and pteridine. So depending on various factors, folic acid can exist in multiple forms, as we said. One thing that might happen to this moiety is that extension of the glutamic acid side chain with multiple glutamic acids linked to it. This is what we call the polyglutamyl form of folic acid. In the next slide, here you will see the various available forms of folic acid in the bottom, based upon the various substituents that occur basically on the N5 and the N10 molecules of the pteroyl moiety. And again, as you can see on the right here, this is the N5 and this is the N10. And here is where multiple glutamyl groups can be added to form the polyglutamyl forms. So if you remember, methyl tetrahydrofolate is the one which is important for the formation of vitamin B12. In contrast, 5,10 methylene tetrahydrofolate is the one which is important for the synthesis of DNA and is the cause of megaloblastic anemia when it is deficient. And you have other forms that are participating in other processes in the body. Folic acid is the form that is used in treatment with methotrexate when you want to prevent the toxicity of methotrexate when you give high dose methotrexate. So in any way, the folic acid, therefore, the pteroyl glutamic form is the form that is given medicinally, and it can be converted to the various forms. Our food contains a lot of polyglutamates and different forms of folic acid, and they are absorbed in the small intestine in the form of methyl tetrahydrofolate. So it is reduced, these forms are reduced and methylated to methyl tetrahydrofolate, and this is the form that is presented to tissues and eventually gets converted to tetrahydrofolate, in this process generating the methyl vitamin B12. The methyl tetrahydrofolate itself undergoes an enterohepatic circulation, and alcohol can interfere with that. And it might be one of the causes of megaloblastic anemia, one of the rare causes. As we mentioned early on in this lecture, alcohol can cause macrocytic non-megaloblastic anemia, but it also can, in certain situations, cause megaloblastic anemia. Tetrahydrofolate can have multiple glutamates, and these are the storage forms of folic acid. And we said that it has the methylene, 5,10-methylene form of it, which is the active form for synthesis of DNA. Where do we find it? Well, source folic acid, as the name implies, from folia, from leaves. It is found in leafy vegetables, fresh green vegetables. It's also found in the liver and in some fruits. Note, however, if you cook these foods, you might lose a lot of the folic acid. Now, what is recommended is around 400 micrograms per day. This is the need for folic acid. In pregnancy, you will have to increase this intake, and that's why pregnant women are frequently administered folic acid as supplements prophylactically, because deficiency of folic acid, as you will see later, not only causes anemia, but in pregnant women especially, it can cause abnormalities in the fetus, neural tube defect. These are some of the foods that are rich in folate, as we said, leafy green vegetables, asparagus, broccoli, seeds and nuts, okra, bamia, cauliflower, etc. What causes folic acid deficiency? Well, the deficient intake, as obvious, is one cause, or increased demand, as you would get in pregnancy, during infections, chronic infections, if you have hemolytic anemia. Malabsorption, problem with absorption in the small intestines. Again, there are certain systemic diseases that can cause folic acid deficiency. And remember that vitamin B12 can trap folic acid in the form of methyl tetrahydrofolate, and which is the one which gets trapped there, and you get deficiency of folic acid when you have vitamin B12 deficiency. Deficiency in the sense that it's not available for the various reactions. Multiple antifolate drugs exist, and you have studied many of them, such as methotrexate, trimethoprim, pyrimethamine, and also you will realize later that some anticonvulsants like phenytoin also have an antifolic effect and can cause anemias. The results, as you would expect, megaloblastic anemia, which you cannot really distinguish from that of vitamin B12. So same presentation, same signs and symptoms, of course. But neural tube defects have been reported in the fetus. One of the problems with folic acid deficiency, associated problems, is the development of hyperhomocystinemia. And this in itself is a risk factor for coronary artery disease and peripheral vascular disease. And it could be a compounding effect for people who already suffer from these diseases. But you don't see with folic acid the kind of neurological deficits that you see with vitamin B12 deficiency, because it's thought that those are related directly to the deficiency of the methyl vitamin B12, which can still happen even if you have some folic acid deficiency. These are the neural tube defects, an example of what is called myelomeningocele or meningocele, with incomplete closure of the neural tube defects, incomplete formation of the spine, causing spina bifida, sometimes anencephaly, and lack of development of the brain. So the question arose, should foods be fortified with folic acid to prevent these birth defects? What do you think the answer should be? What is the treatment for folic acid deficiency? there are a lot of folic acid preparations that one can take. Oral intake is appropriate. Even if there is malabsorption, you can give enough doses that can still be absorbed enough to remedy the situation. Features of megaloblastic anemia, whether it's folic acid or vitamin B12, usually the onset is slow, it's insidious. You get the normal signs and symptoms of anemia. Sometimes you might get some mild jaundice in addition. It's not primarily a hemolytic anemia here, but because of the ineffective erythropoiesis in the marrow, there might be a release of hemoglobin, breakdown of hemoglobin, and therefore you might get some increased bilirubin, unconjugated bilirubin in the plasma. You get the other signs and symptoms which you encountered also in iron deficiency, such as glossitis, angular stomatitis, or chylosis, and uh, this is the pallor that you can see in a patient. These glossitis and the chylosis or angular stomatitis on the mouth. Lab data will show macrocytic changes, also oval macrocytes. The decrease in reticulocyte count for the anemia. In anemia, you expect to increase the reticulocyte count, but when you have a defect in the synthesis of the red blood cells, of the development of the RBCs, you will not show reticulocytosis in contrast to what you see with hemolytic anemia. The white blood cells and platelets might decrease as well. You have a problem with DNA synthesis, remember. You get the hypersegmented neutrophils, as we discussed earlier. A busy marrow, hypercellular marrow trying to compensate. Large erythroblasts, giant metamyocytes, and there could be increased LDH in the serum. And we said also increased bilirubin in the serum, in the plasma.